introduce Professor Tríona Lally here today. Tríona is a lecturer in Biomedical Engineering in Trinity College Dublin, and she's the current director of the Trinity Centre for BioEngineering. So the title of Trina's talk today is Made to Measure, 3D Printing of Paediatric Stents. So go ahead, Tríona, when you're ready.
Okay, thanks, Robbie. Thank you for the introduction and the invitation today. It's lovely to be invited by a past PhD student who's now on the Biomedical Engineering Division, as I was many years ago. So it's lovely to be invited by Robbie today to speak. So I'm delighted to join you, as I said, I suppose I don't know exactly the background of the people coming in here today, so I thought it might be useful for people maybe who are at their early stage of the career to even understand my background, a little bit about what I do, before I go into this particular topic that I want to talk about, because just the nature of research, I suppose, we do a lot of different work, and given the time frame, I'm just going to really talk about one particular study in detail, but I'd like to give an overview, a little overview of what I do as a professor in biomedical engineering in Trinity College, as I am now. So, and I said, my name, I pretty much go by Tríona all the time, as Robbie said there, but if you're looking for my details or publications or things, it will be under my full name, Tríona Lally. So, a little bit about my background. So I started out doing mechanical engineering in University of Limerick. And this was really because, although I loved biology, maths and physics, and that was what led me into engineering to begin with, I probably, if there had been a biomedical engineering degree, would almost certainly have chosen it, because I was kind of seeking something like that. But back when I started out, which is a very long time ago now, doing my degree was 1993, there wasn't a biomedical engineering degree, and thankfully now there are many. So then I decided to do mechanical engineering, and during my degree I was very fortunate to have a work placement as part of the degree, and I ended up working for a medical device company which is now, it's in Limerick, and it's now called a Stryker in Limerick, but at the time it was Homedica, and they make prosthetic joints. And so once I got in there, I knew absolutely, yeah, that's what I wanted to do. I wanted to be a biomedical engineer. So that was what kind of set me off to doing then a master's after my degree in by research, again in University of Limerick. And it was looking at fluid flow dynamics. And it was probably my first kind of introduction and development of my interest in computational modeling. So what I did there was I did experimental work as well. So we looked at a lot of kind of bypass junctions and fluid flow in those bypass junctions, but we also used computational models. And the problem there was essentially when a knee vessel, in the leg or down by the knee can become blocked, it can be bypassed using a graft. And these grafts, depending on their geometry, can be successful or not. And we really wanted to look at, well, given the geometry, what was it about the geometry? How could we optimize it to get better blood flow in order to prevent that bypass from blocking up again? And so it was really interesting. We used lasers to look at fluid flow and kind of characterize the fluid flow in experimental setups like this of bypass junctions. So you can see here of this kind of complex arrangements, and we changed the geometry, and then we created these in computational models as well, and we were able to change it more freely there. And I suppose I really got hooked on this idea of kind of solving biomedical problems using engineering techniques. So from there, although that wasn't really the plan, my plan was always to go to industry, where it would be, I thought, at the cutting edge of developing medical devices. After the research masters, got an opportunity to do a PhD in Trinity College Dublin with the current provost of Trinity, Professor Patrick Prendergast, and linking with the company. So that was what really, I suppose, made it attractive for me. It was working with the company Medtronic in Galway at the time, and it was looking at this whole area of stents, intravascular, these little metal mesh structures that are placed inside arteries to open them and keep them open when they've become blocked. And so what I did as part of this PhD was I tested a lot of arterial tissue. I used that information then to describe computational models, and I looked at how we could improve stent designs. And this is way back, I finished in 2004. And you will probably see, and even know from the title now, this work that I did as part of my PhD has had a big impact on the direction I have gone in my research as an independent academic. And it's been an area of keen interest always, this whole area of intravascular devices and stenting, and of the mechanics of vascular tissue. So that was, I suppose, following my PhD, I set up my own research group and became an academic and was appointed as such in Dublin City University. So lots of universities around Ireland at this stage. And I was there for 11 years as a lecturer and then a senior lecturer. And there, as I said, I started my kind of independent research career, had a number of PhD students working with me and postdocs, and carried out a considerable amount of research before moving over to Trinity College then in 2015. So I was lucky to get a large grant, a European Research Council grant, which can give you the freedom to go anywhere really. And with that grant, I came back to Trinity to work in the Trinity Centre for Biomedical Engineering, because I had, I suppose, maintained collaborations with that centre, and there is a real critical mass and fantastic friends and colleagues that I work with there that help, I suppose, to move forward the research that I do. So I've been here in Trinity since 2015 as a professor in biomedical engineering. So that's a little background on how I got to be in this position, but also now a little bit about what I do in that role. So as you can imagine, I can't go into detail on all these things. So this is a real whistle-stop tour of kind of the different things that we do. And then I'll go into detail, as I said, on one. So a lot of what I do is testing of biological tissues. So everything from animal arteries, veins, to human tissues that we extract in surgery, cerebral arteries from the brain, as well as carotid arteries and carotid plaques from the neck. And we mechanically test these to better understand these tissues, to understand how med devices, when they go into these tissues, might actually interact with them or even induce further biological reactions. Because I suppose that's the difference between biomedical engineering and other engineering fields, is that we're dealing with a dynamic environment. Everything we do within the body, there's going to be a biological response to, and understanding that will help us to improve the devices that we implant. Other areas that I work in is in tissue engineering. So many of you have heard this term, and it's a really growing area. And here you can just see even just one simple, like one little picture here. This is a porcine artery we take from a pig. We take all of the cells off it using a particular process to remove those cells that actually passes reagents through the vessel and selectively removes the cells. And then we can repopulate that vessel with cells from humans or other animals per se, but ideally being humans, so that we can use this now as a bypass graft rather than using synthetic materials, and they tend to have greater success. Other areas that I work on are in looking at heart valves. This is project work that we've had ongoing now for 5 years with Boston Scientific, particularly looking at why these valves, which are bioprosthetic, they have leaflets made of a biological material that comes from the outer surface of a cow heart. And these leaflets, their lifetime is limited in some cases by the fact that this valve opens and closes and opens and closes. Like the mechanical loading that's placed on those leaflets means they don't always last as long as we need them to. So we want them to last like, much longer than 10 or 15 years. And in some cases, they only last 5. So we look at the mechanics of this tissue and see what can we understand about the underlying structure that might govern that lifetime. And better understand it, and also create computational models to help these companies and ourselves to better design these devices so they will have greater longevity. So that's, again, just an idea of some of the things we do. Something else I want to kind of mention, because it's very novel within our group, is we look at non-destructive characterization of tissue. So the structural characterization. We try and look at the underlying structure of tissues in non-invasive ways that might help us to better understand disease progression and for ultimately, I suppose, diagnosing these diseases. I'm just going to mention one type of imaging that we do in our group that is quite unique, I suppose, to our group, and this is called diffusion magnetic resonance imaging. So many of you have heard of magnetic resonance imaging. This is an imaging technique for looking at tissues, particularly like their anatomy, so you'll get an idea of the shape, size, how blocked they are, things like that, with this imaging technique in a non-destructive way. But there are other types of magnetic resonance imaging that can track water molecules, so the diffusion of water molecules within tissue. And this can help us to understand changes in the underlying structure of the tissue, which might be indicative of disease. So within our group, we use this technique, diffusion magnetic resonance imaging, to track water molecules in arterial tissue. And this image here just shows you, if we had water molecules, so you can imagine those red particles there, and if we allowed them to diffuse in every direction in a material that we call isotropic, is the same in every direction, they will go everywhere. So you just see, that's your standard kind of Brownian motion. They'll move your diffusion, your particles are going everywhere, diffusing out. But what if we have track water molecules within tissue that has a structure? So imagine arteries, they have fibrous structure, they have cells arranged in a particular way, and we track water diffusion in those tissues. What we see is that the water will diffuse along the underlying structure. And therefore, if we track water, we also track the underlying structure. And so this can be very informative, as I said, for kind of early stage disease. Now, tractography, this tracking water molecules and kind of modeling it, has been widely used to date in brain fiber mapping, so actually mapping the neuronal tracts in the brain. And it's been very successful. Here you can see you're kind of looking at tracks and seeing those things like brain damage or injury to the brain. You might be able to track that by looking at this tractography. It's also been used for tracking cardiomyocytes, the cells and these fibers in the muscle fibers in the heart, and again, enabling you to see maybe damage in the heart following a heart attack or cardiac infarction. But we've also looked at it to see what can we use it to understand changes in the underlying structure of arteries. And we've taken tissue from the neck, which is prone to blockages, areas that we will see tissue grow over time that can actually preclude blood from going to the brain and can result in things like stroke. So we've looked at human tissue that has been extracted from cadaveric tissue. So this would be from bodies donated to science through the Royal College of Surgeons in Ireland. And we've looked at using this imaging technique to track fibers or the structure, underlying structure of this tissue. And we've also looked at it for when we remove in surgery, in a procedure where you do have these blockages in the neck, you can have a cut made down. It's a procedure called carotid endarterectomy. It's a surgical procedure. The plaque is removed from the artery. And in this case, a cut will be made down the neck opened up, a cut into the carotid artery, and this plaque, this blocking calcified and fatty and fibrous tissue will be removed from the artery to allow blood to flow again. And when that's removed, normally it will be actually disposed of, but we get that tissue and we can also track fibers within it using this magnetic resonance imaging technique. So I'm just going to show you here some of the tractography that we obtained. So there on the left is a vessel, so an artery taken from a cadaveric artery taken from a body that's looking at the tracts within that carotid artery. And you can see that there is a clear underlying structure to it. Whereas on the right-hand side, you can see the disorganization that we see when we track fibers within a plaque. And this is something that happens over time. So our magnetic resonance imaging technique might be able to track how we go from here, this organized structure into a very disorganized structure, which is very indicative of disease, and where there is a real risk in this disorganized structure that we will have failure, parts of that plaque break off, flow with the blood, and cause blockages in the brain, which will result in a stroke. So that is one of the techniques we're looking at very closely now and moving towards trying to bring this into in vivo applications. So, as it is, we use it ex vivo, we use it on tissue harvested and in the lab, and it informs us on how disease progresses and what it looks like when it does. So that's just a couple of examples from our lab of some of the things that we are doing. But as I said, I wanted to focus, I suppose, on one particular study today, because as I said, I can't go into detail on all of these different studies, but it's nice to show you as well the diversity of what we do as an academic. We're not working on one project, we're working on many projects at the moment. I have about 10 different projects ongoing, all in this cardiovascular space, but in different areas and looking at different research questions. So that makes it a very interesting job. But the study I want to show you today in a bit more detail is one around, as I said, 3D printing pediatric stents. And this is a study that's very close to my heart, if you'll excuse the pun, but it is because the way this came about, even this particular research, is interesting. Because there's a number of people working on this team. It's a very collaborative project, and we are continuing to grow this team and bring people on board and get funding in other areas for this work. But I suppose it's worth noting how it started out. So my sister, her daughter, had a heart transplant, and while having heart transplant, they're not carried out in Ireland, actually. They're carried out in London, children's heart transplants. There's not enough children having heart transplants for them to have full expertise to do the procedure, the surgical procedure here. But they are supported here. So my niece would have gone over to London, she would have had her heart transplant, and she would have been kind of hospitalized here and her case managed here until she was ready for the transplant, and then returned back after her transplant as well for her aftercare here in Crumlin. And it was through that whole process, I suppose, seeing my sister and her daughter go through this, that in Crumlin, that I started to get really interested and go, well, what could I do as a biomedical engineer? And I got talking to the consultant that had been treating my niece here in Ireland, Professor Damien Kenny, who's here. And we realized there were lots of areas where the expertise I had on areas like 3D printing and 3D printing of geometries could help them with preclinical testing, but also in 3D printing medical devices. And so this is where this project came from. It has had a number of people working on it, as I've said. So we work with Professor Damien Kenny in Crumlin. We see procedures and we go out to work with him and see what the need and the problem is. Then we've had a number of people. So Dr. Orla McGee has been a postdoc working on this project until recently, where she went over to Canada to continue her research career. We've had a internship student. So this is interesting. A third year student, Celia Hughes, came to work with us for a summer and contributed hugely to the work. And then she is now a PhD student in the lab a year or so later. Which has been great. We work with a group in Birmingham, University of Birmingham. So Dr. Parastu Jamshidi and Professor Motaz Atala are experts in using powders to 3D print using this technique called selective laser melting, as I'll show you shortly. So they are experts from a material science and metallurgical point of view, whereas we are more on the vessel side, the geometry, as I'll show you, and the testing. And in particular, we have a lot of expertise in Trinity on printing what I'm going to show you today, titanium stents, but in Birmingham, their expertise extend to shape memory and superelastic materials like nitinol, which is where we're currently progressing into. And this is why this partnership works so well. So this work as well, I should point out, was funded, the earliest work was funded by the National Children's Research Centre. It's notable that this is a charity and it has funded this work. And I'll point again to why it's so important that research like this is done in research labs, as well as I suppose that we be aware that in many cases it's charities that fund this, because that it doesn't have a huge commercial benefit or commercial potential right now because of the numbers. I will say we are continuing this work, which is great. So another PhD student started in my lab just last October, funded by the SFI Centre Amber, to continue our development into 3D printing of pediatric stents. And I've also had recently a postdoc start to continue some of the preclinical models, Dr. Kira McKenna. So she's in our lab now. So here, as you can see, it's growing, and we're continuing to do the work. What I'll show you today is mostly work that has been completed by the group shown here. So highly collaborative. So what is the problem we are trying to address, and that was presented to me by our consultant, Professor Damien Kenny? Well, it's this area of aortic coarctation. Well, aortic coarctation is a constriction of the aorta. So you can see here, the aorta is the largest artery coming out of the heart. It'll deliver all of the oxygenated blood. So all of the oxygenated blood of your body that's going right up to the top of your head, to the bottom of your feet, must go through the aorta first. And then you can see all these branches are what deliver it up into the head, and then subsequently, and the descending brings it down into the stomach and down further into the legs. So if we have a constriction, here you can see a very dramatic one in this large vessel delivering all that oxygenated blood, it will limit not just the quality of life of an individual, but ultimately also their lifespan. So aortic coarctation can present itself very early. In fact, it can be detected in the early fetal scans, in the 20-week scan, and might even be treated then in a baby, in a neonate, very early on. But it can also, if it's not serious enough or enough of a constriction early enough, it might progress and not be picked up till the child is a little bit older. But what we see is that it occurs in a limited number. So it is a congenital defect. It's one of the many congenital defects that occur, cardiovascular congenital defects that occur in children, but it's relatively small numbers in terms of 3 in every 10,000 live births will present with a coarctation. But when it does present, it said it's very serious, and it will limit the lifespan of an individual, both their quality of life until that point, but their lifespan also to about 35 years of age. So very serious if not treated. So some of the treatment approaches at the moment would be in neonates, in early treatment, it's surgery predominantly. So you can see there's lots of different approaches for surgery here, everything from just cutting out the constriction and then doing an end-to-end anastomosis, or like suturing of those ends together, to putting in cuffs or grafts or little patches and flaps. So what we see here is, this surgical approach here being applied. As I said, this is very successful if done early on neonates. But what we see there we go. What we see that when this presents in older children, so maybe, in anything from a 2-year-old upwards, it'll generally be tried to be treated with a minimally invasive approach because the surgery will be risky for the individual. So a minimally invasive approach that we can take to opening up that constricted aorta is to use a balloon. So bringing a big long catheter, a long flexible structure, goes up from the femoral artery, so down at the groin, right all the way up into the aorta, and a balloon is expanded in that location. You can see it here to open up the vessel. And typically for greater success, when that balloon is expanded, it'll also expand this little metal mesh structure, the stent, at the same time to keep the vessel open. Because when you deflate that balloon and remove it, often that constriction will recur again unless you have something keeping it open, scaffolding it open, and that's essentially what a stent does. So the little metal mesh structure will be in there, keeping the vessel open and restoring blood flow. So why isn't this like wonderfully successful, and why do we need to print stents to overcome any limitations? Well, the reason is really because at the moment, these stents that are being used for aortic coarctation treatment in children are not designed for that purpose most of the time. Because again, the numbers are small, that 3 in 10,000. Companies won't develop them. They won't put the research and development time and money into producing stents that are designed particularly for that treatment application because the numbers are so low. So given the limited stents that are available, the products that are used in these surgeries are often not very well suited. In fact, most of them tend to be designed for the likes of the biliary duct. They're not actually even designed, for pediatrics or for the artery they're being placed into. This can result in clinicians feeling they don't have the tools they need, and they end up having to modify some of the technology that they have, some of the stents that they have access to. So they use them, as the term is, off-label, which doesn't guarantee their success really, and they end up maybe cutting them or shaping them or even hand crimping them, so crimping them down to go into the catheter that will deliver them. All these things can have an impact on the long-term success of the procedure that faces this product. The other thing is, if you look at the particular geometries that we see aortic coarctation within pediatrics, they tend to be very specific to an individual. The coarctation is not necessarily going to occur in the same place. The geometry of the vessels can be very, very different. So here you can see just two examples. These are images that were taken of children who were presenting, needing treatment for aortic coarctation in Crumlin. The one on the left is from a child who's 5 years old, and you can see that the constriction is in the arch of the aorta. Whereas here on the right-hand side, this is a child of 12 years of age, and you can see the constriction is to much greater extent in this vessel, and it's down here in what is the descending aorta. So these two present completely different challenges. Here we have a curved vessel. We're going to need to put a stent in there that's going to conform to that vessel and not straighten it too much. But you can also see that there are many of the main branches going to the head and neck coming off this vessel could be occluded by placing a stent in there if that's not considered in its design. Here on the right-hand side, we can see that this one here going down into the descending, again, high level of curvature. If you put in a very rigid stent in there, you're going to straighten that vessel, you're going to dramatically alter the blood dynamics, and that can have a big impact on downstream effects. So these are, by their nature, complex, very patient-specific, and we have to remember that these vessels are also growing. So stents going in here really need to be able to expand over time to allow for that growth. So all these place challenges on the stents, and we really don't have the stents available that account for these complex and patient-specific characteristics. So it was thinking about these things that we thought, well, could selective laser melting or 3D printing enable us to create products that would be more bespoke, suit those complex geometries, suit those individual patients? And here is just a video taken from SLM Solutions, Selective Laser Melting Solutions, showing you an idea of what do we mean by this? What do we mean by 3D printing? Now, many of you have heard of it, and many of you have even seen products that have been 3D printed, such as, maybe from what's called fused deposition modeling. So if you've seen plastic products that have been 3D printed, they've been laid down layer by layer, just like we do here with the metal. But it's quite a different process in the sense that there you're melting a plastic and extruding it and then laying it down in a layer. With selective laser melting, what we're talking about here is you're actually welding with a high-powered, very high-temperature laser spot points and then moving through a powder and continuing to weld those spot points until you've created a layer. So this laser beam is hitting this thin layer of metal powder, selectively welding the particles together. And here's just an image showing you that. So this is like, see this one layer being laser welded together, and then a powder layer comes over again, with a little kind of wiper over it, there we go, and then that powder layer can be melted again on top. And so it's layer by layer fashion, as we can see here, that this build platform be lowered and a new layer of powder placed on top. So that's how the 3D printing technique works for metals. So we wanted to see if, well, could we actually generate to begin with 3D printed patient-specific stents using this technique, this selective laser melting technique? Was it feasible, for one thing, to create the kind of geometries needed for these types of stents, or this type of application? Well, before we could do that, we needed to understand, well, are there any limitations to printing in selective laser melting approaches? And actually there are considerable limitations when you think about printing something in a layer by layer fashion from this metal powder. In fact, they have to be what's called self-supporting. You can imagine if you're going up in a layer, you can't just have a big overhang because it'll fall down. You need to either support that with something in the higher levels that you've printed, but you do need to consider what are the limitations to the design of a stent that can be printed using selective laser melting. So these are some of the kind of criteria that have been identified to limit the designs that stents or any 3D printed metal structure can have. And this is from Demir et al. That they identified, well, we really need to have angles greater than or equal to 45 for it to be fully supported. Any less than that, it's essentially going to collapse on itself. You have an overhang limit. You can overhang a little bit, but only by about 1 millimeter. And if you support on the other side, you can go a little bit wider, but you need to keep it less than 4 millimeter if you're bridging from one side to another. And when it comes to gaps, actually, you do have to have gaps of large enough size, because otherwise you'll fuse things together. So the gap that you can have must be greater than 0.3 of a millimeter. So considering these types of constraints that we have on the selective laser melting process for printing stents, we can see that maybe a design such as that on the left, which is typical of a more complex stent design, is not really achievable because it's not self-supporting. It has overhangs that are very long. So you can see here, these would be the overhangs when it's printing in this direction. We would have these overhangs, and that wouldn't work. So we have to print something more like this, a honeycomb type structure, because we keep everything there at angles of 45 or greater. So knowing this, we said, okay, let's try and design some stents for additive manufacturing. So we said, let's consider that, yes, okay, the honeycomb structure is really what we can work with in terms of printing stents. But surely there are ways that we can maybe post-process this. We can carry out other techniques after our printing to maybe end up with something that is more flexible, as I'll show you. So something like this on the right-hand side, which is what we're calling our novel design, in this case, which is an open cell structure, which, as you can see by just looking at this, has overhangs that are way past 4 millimeters and really doesn't conform to our design guidelines. So we had to think a little bit outside the box and go, well, okay, we want to 3D print, but perhaps we need something to modify what we can deliver with 3D printing to get what we ultimately want for delivering the best treatment for individuals, or at least to give us flexibility in design. Can we have these open cell structures? So we had other issues to consider as well when it came to 3D printing, is that there are printing defects. The way that that laser works, we end up with a lot of this debris or particles attached to our struts. And that can have a big impact on things like, the surface finish, on how it interacts with the artery. And it's really not possible to place a stent in that would have all of this debris or dross on the 3D or as print structure. So, and here you can see this is a scanning electron microscopy image of that of an as-built strut, and you just get an idea of what those kind of defects look like. All these little particles that attach, that haven't been fully welded, they're just powder particles attaching when you're laying down those layers. So how might we get rid of all of this defect and poor surface finish, as well as try and give us flexibility with design, even though we're constrained by 3D printing rules? Well, let's see, can we use something like chemical etching to remove these defects and remove material as needed? Well, the type of chemical etching you'd have to use for the stents that we're printing here, which are titanium stents, requires you to remove titanium, would require you using hydrofluoric acid. And hydrofluoric acid, when you etch away, will produce a lot of hydrogen. And when that happens, that can result in actually cracking of the structure, having that hydrogen embedded in it. So we have to add nitric acid as well to remove that hydrogen, as you can see here, and turn it into water and nitric oxide. So that's what we use. We use now a chemical etchant solution that has hydrofluoric acid, nitric acid, and water to ensure that we can remove material and also potentially give us flexibility in design. So I said, what can we do? Well, if we can remove material, then perhaps we can put supports in and have honeycomb type structure, as you can see here, but ultimately through etching, remove those supporting structures. So you can see here, so we make some of the struts here thinner. So really they're only acting as supports. They're not actually part of the stent design. We expect to remove these in our etching procedure. So that's how we will go from our as-built here to keeping with additive rules to getting to a much more complex stent structure. So let's see how that works. So essentially what we want to do is then we print stent designs with additive manufacturing rules. So we do our printing. We then take those stents. This is our simple honeycomb. We have the SH, so we'll keep this kind of acronym throughout. So SH is our simple honeycomb stents as printed. These have quite large struts. Actually, they're about 0.5 millimeter, as you will note from some of the previous schematics. They're about 0.5 millimeter thick, which is very thick for a stent. Typically, stents would be around 100 microns or lower, even 50 to 100 micron strut thickness. So we're going to use the etching to bring that down, get a better surface finish, but also to give us more flexibility in design, to enable us to remove supports that otherwise, if you try to cut those supports away, you would break the stent. So you can't really do that. So this etching procedure is a way that we can do it effectively, or at least we hope. And I will show you here how we achieve that. So I suppose we have two approaches here. We etch away to thin the struts and we etch away to remove supports. And we have actually two different etching times here for these just to achieve those. And you'll see that has a big impact as well on the performance of the stents. So here is our Three stent designs are simple honeycomb etched. So it's essentially this design with uniform thickness put into an etching solution to thin the struts and to give us greater flexibility as well as improved surface finish. And here our novel design etched, we just etched to the point where we remove the supports, and we see if that'll give us this open cell structure. And you can see that it does. So let's evaluate these a little bit then. Have they worked? Do we get reduced strut thickness? How does that affect our surface roughness? Can we carry out, for example, crush testing then to look at the performance, the radial stiffness of the stents, and are they suitable for going into a coarctation and opening up those vessels? And then the final thing I'm going to show you is this inverse FE that we do in order to ascertain what is the actual mechanical properties of the titanium when we print it, because we cannot assume it's the same as the bulk properties of titanium. 3D printed titanium or any metal when printed can be very different from a bulk as cast material. So we look at, I will show you a technique that we use called inverse finite element analysis that can enable us to calculate or work out what the stiffness of our as print material is. So let me go through each of those, and then I'm going to show you how we bring this into the real vessel geometries to help us optimize sets. So looking at our printing and etching results, what you can see here is from our simple honeycomb to our novel etched and to our simple honeycomb etched. Even just qualitatively looking at these initially, what you can see is that if we try to bend the ends of them, just look at them in terms of what's their bending stiffness, even qualitatively, you can see the open cell structure. Even intuitively, you would see that the open cell structure should allow greater conformability to a curved vessel. Whereas these two geometries, even when we thin the struts, it doesn't give us that conformability because we have closed cells, all connected, which gives that real rigidity across the stent. So it's only in opening up the cells that we can get that flexibility. So it is feasible once we etch to get these open cell structures and keep the additive rules. So we were quite happy with that we could enable this flexibility in design. Going from there, we then thought, well, let's look at, well, is this stent likely to have any structural integrity after etching? Will it have the kind of strut thicknesses that are similar for and surface finishes that are typical of a commercial type stent suitable for implantation? And we did that by looking at microscopic measurements of the stent. So you can see here just different microscopic measurements from this one is actually thinner. As I said, the time to thin out the struts, you can see we got thinner struts here than we did with the novel etched design, where we just removed the support struts. And you can see that they are quite uniformly etched. And so we could get this novel design and we could reduce the thickness. So let's look at some of the thicknesses here. So I just want to appreciate these because later this has implications for now the radial stiffness of these stents subsequently when we test them. So you can see we've gone from about 0.5 millimeter, so 500 microns or 0.5 millimeter in the as print stents, which are quite easy to print because they're nice and thick and that's easy to print, but they've again a lot of dross around them, a lot of particle attachment or defects. Once etched, I'll show you the surface finish now in a moment, but we can get down to much closer to typical commercial stent design, so down to around 120 to 150 microns. And so that is possible, and we can even go potentially lower, and we are working on that at the moment. But with the novel design etched, once we remove the struts, we had thicker struts, but again, we could have etched for longer in order to thin those out further. But we can definitely remove the struts to give us this open cell structure design. And here we can see just some closer up images of both the scanning electron microscopy images of our as print with all those defects and all of that dross attached and particle attachment. And here you can see the nice surface finish that we can see even qualitatively here that we can get once we etch. But let's look at this in a bit more detail. We can see that the thicknesses definitely have reduced and we have the thinner struts for this simple honeycomb etched, as I said, around the 150 micron, whereas around 200 microns from the novel design etched one. And that's compared to what it was in as print, which was just over 500 microns. But now we can see that this controlled reduction in strut thickness can be achieved, but we can also achieve at the same time, which is really beneficial, good surface finish. And we used a technique called white light interferometry to look at the surface finish of each of those stents. So before, the as print with all the defects, and you can see that here, all these peaks represent little particles that are attached onto the surface. Here we can see once we etched for a period of time to remove the struts, which again reduced the strut thickness somewhat, but wasn't as long as for the simple honeycomb etched. We do get improved surface finish, but we could probably etch for a little bit longer to improve that again. And here you can see when we do etch for longer, those thinner struts result and we get a very good surface finish. And some of those metrics there, you can see our mean roughness is down at the order of only less than a micron when we etch for that simple honeycomb etch to get down to strut sizes of around 120 to 150 microns, which again would be typical of more suitable from a commercial point of view or from a real application point of view. That's the kind of strut sizes we want. So chemical etching can definitely, it reduces surface roughness and makes these more suitable for implantation. So the next stage is once we had our white light interferometry, we'd reduced our surface roughness. We now wanted to see, well, would these stents have the appropriate radial stiffness to implant and perform a coarctation, so open up a vessel and keep it open. And then we also wanted to see, like, how did that compare for the as-built and those that were etched. Then we're going to create that FE model, as I'll show you, to do this inverse FE to work out what is the stiffness of the titanium that we use when it's printed. And then I will show you the patient-specific models. But here's just an image now showing you those stents being crushed using a parallel plate testing setup where the force that's being applied to the stent is measured as the stent is deformed here. And doing this, we can get out from that what is the radial stiffness, what's the force here needed to crush those stents, excuse me. So here, the simple honeycomb, the thickest strut stent, as you can imagine, this one is going to have a greater radial stiffness. In fact, it takes around 100 newtons to compress that stent to 3 millimeters. Whereas when we look at the novel design etched with those thinner, around 200 micron struts, we see that it takes about between 5 and 10 newtons for each individual stent. There's some variation. Again, this was because there was some variation also in the etching, and you saw that in the strut sizes. And the same with then the simple honeycomb etched, which went to the thinnest struts, needed the lowest force to compress it. So it had the lowest radial stiffness, so the greatest compressibility, this simple honeycomb etched. And again, a little bit variability again, because there was variability following the etching in the strut sizes. And that was again, just more, we need to do more control studies on the etching. As I said, this was more of a feasibility study. Is it possible to do this? And can we use this technique? So it's worth noting this lower one here, actually one did have a fracture, but that was just one out of all of the stents that we printed, and in fact had a fracture on that strut. And it does impact the radial stiffness, of course, and it will have an impact, but overall we found the etching was very successful for reducing the strut size, increasing that or improving the surface roughness, and also controlling the radial stiffness. So if we want something more conformable, less stiff, we can definitely do it by etching away. Okay, and just you will note that the order here, there's a full order of magnitude almost difference, or 2 orders of magnitude going from here up to the simple honeycomb with that thicker strut. And that is down to the thickness of the struts. As you said, around 135 here, 225 here average, and up to 560 microns here on the thickness. So our reduction in radial stiffness can be achieved, and it very much just depends on the strut thickness, which is controllable through etching. So then we went on to see, well, actually, is the, when we print the stent, what is the stiffness of the titanium in its as print state? And that's quite a difficult thing to do. I mean, you can make small little coupons and test them, but again, it is very difficult to look at the impact then of even things like the angle that you print at and so on. So we decided to use what's called an inverse finite element approach, where we create a finite element model, which exactly replicates what we do experimentally. And we can iterate then using a minimization approach of the difference between what we see experimentally and what we see numerically by changing the Young's modulus of these stent designs that are created in our finite element model. So we crush these in the same ways we have done experimentally, and we determine what Young's modulus do we have to put into the stent here in order to replicate what we see experimentally. And that essentially tells us then that that must be the Young's modulus, or the stiffness of the titanium in its print state. So we did that for all those different stents, those 9 different stents, so 3 from each of these geometries. And we were able to get out results that we could match our Young's modulus. So these kind of colored curves here are showing you what is the Young's modulus calculated to be when we match those experimental curves from those 9 different stents. If we replicate them in the finite element model, what Young's modulus is needed in order to get that same deformation seen experimentally. And here is a table of that. So for the 3 different simple honeycomb, the 2 simple honeycomb etched, because one had a fracture, and then the 3 novel design etched, we were able to calculate what would be the Young's modulus needed to create that deformation we saw experimentally. And what you will note here, particular note, is that we have, okay, for the simple honeycomb etched and the novel design etched, while there is some variability, we have close to, up around 100 gigapascals for Young's modulus, which we would expect for something like a 3D printed titanium to be lower than the bulk or as cast material, but not to be as low as this. I mean, to see a 31 there to 35 gigapascals as a Young's modulus, this is very strange because this would be very low for titanium. And the realization here is that the reason we're calculating these to be so low is because of that dross, that particles that are attaching onto the struts in that as print state that are essentially overestimating the thickness of the struts, but that's not really load-bearing material. So that with that overestimation of thickness, essentially you will underestimate the Young's modulus. And this is again just reaffirming the real importance of etching the stents to get a better surface finish, but also to remove non-load-bearing material, which you might think is going to take the load in that stent and which would make you think you have a thicker strut in that case. So all of these are much more representative of what the print, the titanium, the load-bearing titanium as printed is in terms of its Young's modulus. So defects, these non-load-bearing defects can result in that underestimation of the stiffness of the stent, and this could have big implications when designing stents, so it's another reason to make sure to post-process this through etching. So finally, once we had done this FE, inverse FE, to calculate the stiffness of the stent, we were in a position that we could actually use our geometries that we obtained clinically to create stents for a particular application, or bespoke stents. So how do we go about that? Well, we now here, this is just showing you for one example. As I said, we have other geometries also, but this is for that 5-year-old patient presenting. We were able to get a CT scan of that vessel where the stent was going to be placed up here in the coarctation. And we were able to create, using different softwares, we were able to create a mesh that we could use in that finite element software, that we could virtually simulate a stent being implanted into this particular vessel. What's also interesting actually is we're not just limited to creating that virtual model. We can also print that physical model using other printing techniques. So using stereolithography, which is a photo cross-linking technique, we can use an elastomer as shown here to print the vessel itself, keeping the geometry, that patient geometry. And then because we've printed the stent itself, we can implant that stent on a benchtop. So in a benchtop model, we can, in a way, as we say, pre-clinically ascertain, does that stent suit this coarctation? Is it going to open up the vessel in the appropriate way? And therefore, is it suitable for this application? So that's a real bonus that we can do that, because you can physically hold this. Surgeons can look at it, they can plan their surgery and see about where they should best place that stent. But then in the virtual model, we also can now look at that stent implantation. So this is just one of the stents, the honeycomb structure being placed virtually inside that real vessel geometry of that 5-year-old with coarctation, and looking at how that stent will open up that vessel. And also looking at what's great here is it enables us also to actually estimate what kind of loading or stresses might be placed on that vessel in so doing. It enables us to see straightening of the vessels or blocking of any vessels that are emanating from that coarcted region, and to, again, pre-clinically assess, is this the most suitable stent? Are you going to get the best outcome from using this stent, or would a different design be better? So we've been able to use these preclinical models now to test the different designs that we have, the simple honeycomb etched, the as-print stent, the as-print stent. This is actually as it was printed after taking measurements of it, and this is what we thought we were printing based on using our kind of CAD geometry. So would that be the best outcome? So should we kind of modify our CAD in order to get this to be our final printed geometry? And these are all things we can do now. We can use this information to improve the stents that we're printing. This is our novel design etched, and the real advantage here is that there's a branch right there, but we have an open structure there that will not block that branch. So this would be much more suitable for in this coarcted region. And again, we have a simple honeycomb here as printed, but with a different Young's modulus, so we can test that too. We can see what if it has the Young's modulus that we predicted it to have, with that overestimation of stiffness, or what if it has the Young's modulus that we predict as the average for titanium. And again, you can carry out this in a benchtop and validate as well, which is the most accurate. So again, we have a lot of ways that we can use these models. And finally, then here are a simple honeycomb again with the STL with a different Young's modulus. So numerical models enable us to test a lot of different hypotheses, enable us to look at different geometries, see what the outcomes would be in a really cost-effective and efficient way. So we can really work with this now to optimize the stent to suit this particular geometry. And we're not just limited to this geometry or others. We can look at things like the stresses, how much lumen gain we get. So here you can see this is like how much do we expand the vessel? Well, our SHE, they're not as, this simple honeycomb etch design and our novel etch, they have thinner struts, they're not as radially stiff. And so there's consequences for that. That means that the vessels that they are implanted into don't open up as much. But that also means the stresses in those vessels aren't as high. So what this means is that we know there are trade-offs. The more we expand the vessel, the more loading we place on that vessel, and potentially the more we injure it. If we want to expand our stent, or we want to achieve a certain luminal gain, we can do it and minimize the amount of loading we place on it by redesigning the stent and preclinically testing it in this type of virtual scenario. So now we are in a position where we know we have flexibility in the design of stents using our 3D printing approaches, using the etching in particular to etch away support struts and to improve surface finish and to achieve thinner struts that really control our radial stiffness. So now we have a framework or a platform that can optimize stent designs to deliver a bespoke treatment for children that is governed not by, commercial interests, but governed for the best outcome and can be delivered dependent on the geometry, the complexity, and so on. The other thing it's worth noting is, before I finish, is that we are working in parallel at the moment. I haven't all of this done, but we're close to it with nitinol stents. We are etching them. We have created the same type of flexibility in design using our etching approaches. And we are now in a position to start implanting those into the patient-specific geometries and modifying and optimizing their geometry. And the reason we want to use nitinol is, as I mentioned earlier at the beginning, these materials are super elastic. They will actually, you can put them in almost kind of a crimped state and they will continuously want to expand. So essentially nitinol is a way that a stent will continue to want to grow. And so this is really the answer, combined with our framework for designing the best design for the geometry to deliver what is needed in this space, which is bespoke treatment for children that can grow with them and is designed for their particular geometry and their particular disease etiology. So this will deliver the best long-term outcomes for these children. So finally, I'm going to show you a last thing, which is here we now have geometries like this. So this on the left here is a coarctation geometry taken from 12-year-old, as I mentioned earlier, where you can see the coarctations in the descending aorta. One thing that we were very lucky in getting recently is we were also able to get what's post-coarctation. So this was post-stenting. We actually had the outcome, a CT scan, and reconstructed this geometry from it of the post-stenting geometry. And now we're able to actually generate the stent that was put in here, a particular type of stent, an intrastent, and we're replicating this with a virtual model. So we can validate it and we can see, could this stent have been placed better, or could we have designed a better stent for this particular application to even have got a better outcome than was achieved. Now, this is a very good outcome, but you will still notice maybe here in this last one that the stent probably falls short a little bit and straightens the vessel just there a little bit at the end. And that we think we could have overcome by some modifications to that stent. And we're going to show that now with our virtual models for preclinical testing and optimization of stents using the platform that we currently have. And we can print stents that will then be that better design and implant them into our physical model to show we could have had improved outcomes with that stent design. So with that, I'm going to finish. I'm going to thank my group. This is my current group members on one of our many Zoom meetings. And I'll thank all the funding that I've achieved into my group in general, but again, just to remind you, I suppose it's National Children's Research Centre that funded this work along with Amber and some support from the Irish Research Council as well. And I'm happy to answer any questions that you might have. Thank you.
Thank you very much, Tríona. Well, it was a very interesting talk. I see there are a number of questions, so if anyone else has questions, you can pop them in the Q&A box at the bottom. I might start with a bit of a general question. And as this, I guess, technology becomes more developed by yourself and others, how commercially viable do you think it will become? Like, is it likely that companies will adopt this technology, or is it limited to universities and hospitals?
Yeah, it's a great question. And it is one that interests people. I mean, how close are we even to getting these into patients and things like that? I would say with this particular kind of bespoke technology, it's going to be more costly, unquestionably, right? So if you think about this from the point of view of adults getting carnage, I don't see this technology immediately being applied there as it is at the moment, because there isn't the real need for bespoke treatment there in the same way. The vessels tend to be kind of more uniform in terms of their geometry, they're relatively straighter, things like that. It's somewhere like this, and this is why aortic coarctation is in treatment of an area like this, as I mentioned, which is really complex. Do you know what I mean? And there isn't one. You couldn't say, okay, patient is presenting with aortic coarctation. You couldn't say, well, I know where that'll be or what it'll look like. They're so varied, and even the standard aortic anatomy, it often can be variable. Because of the way the child is overcompensating, like kind of trying to compensate. There could be bifurcations in areas there aren't normally bifurcations, so you really have to take account of their individual geometry. Even for planning, actually, for consultants planning the procedures, because of that complexity, having a physical model can even help them to have a better outcome, because the planning can be quite challenging because it's not the standard anatomy, you know. So I think the areas we're going to see this develop, like we're doing here, and where it should develop, is in this kind of where bespoke treatment is really necessary. And I think then it will get more cost effective. And then ultimately, I think it'll be able to be rolled out to the millions of stents that are being implanted for adults, you know, worldwide every year, such that, you know, it will become cost effective for that.
Okay, and there's a number of, I guess, follow-on questions from that. So there's one here. So great talk, thanks. Are there plans to bring these concepts to implantation in children, and how do you balance the time to develop and test with the growth of the child?
Yeah, absolutely, there are plans. It will take time, though. We are still away from, you know, a bit away from being in a position to implant these into individuals, because obviously you can imagine there's a lot more, you know, progress to be done in terms of regulatory and making sure they're safe and they can be produced very repeatably, consistently. You know, you couldn't have this kind of variability that I showed you there in the strut sizes, you know, and say, you'll get approval for putting in a stent like that and saying, oh, it'll be somewhere between 100 and 150 micron strut thickness. You know, this is very much feasibility at this stage, but we are progressing toward that. And towards essentially implantation. But we have a couple of other challenges as well. I mean, things like we would have to work on fatigue life. 3D printed products by their nature, because they're layer by layer, have a reduced fatigue life. Typically, they are more brittle. They tend to be at greater risk of fracture. And something like this that's in the aorta going to cycle, you know, 60 to 80 times every minute, we do really have to look at the fatigue life. We also need to look at heat treatment in order to, you know, kind of remove any pre-stresses or residual stresses that might be in these materials following print. And again, these are things we're doing at the moment. So the next few years, we have a lot of work ahead of us, but we think, you know, we can work these out. We have, as I said, that aspect of flexibility in design was something that would have curtailed this movement into this area. We feel we've overcome that a lot and we're getting a lot of benefits from it. So now we have to move into the heat treatment and we need to move into the fatigue life.
Okay. And then there's, I guess, 3 or 4 questions all centered around the same kind of topic of, as the child grows, is there repeat procedures, bioresorbable stents or nitinol stents that might grow with the child?
Yeah, there's a number of options there. And like, that just been mentioned there, that is one approach, to put in a stent that's bioabsorbable. And so over time, it's reduced in terms of its scaffolding potential and you implant another stent. That's another procedure, so that's not ideal. There's obviously tissue engineering approaches where things can kind of actually, you're putting in a tissue and it's growing. So, you know, I mentioned that earlier, that, you know, we do have some work on tissue engineering, but the tissue engineering aspect is again a surgical approach, because you really have to replace the coarcted tissue and then you're putting in a tissue that will grow over time. And that's still very early days in terms of getting something that you could be sure would grow and would be able to maintain blood pressure while it's growing. You know, the minute it goes in, it has to function as an So what we're going with the approach of this mechanical device that doesn't absorb is with a view to scaffolding open that vessel, but allowing that scaffolding to continue to expand. And that will be achievable with nitinol. Nitinol is this nickel titanium alloy, and there are some other superelastic and shape memory alloys that are emerging. So particularly the superelasticity of them that are titanium based as well, which we are looking to work with also. And these are essentially materials that they try to recover their deformation, but a lot more deformation than is typical with standard engineering materials. So by that, what I'm saying is, in practice, what does that mean? That means if I have a stent crimped down into a small diameter, it's constantly wanting to recover its deformation and get bigger. So once it goes into the artery in a small size, it'll keep wanting to expand. So as the artery expands, it'll expand with it. So that's why we're very focused on applying this technique now to these nitinol powders. And we have printed stents and we have printed these novel design etched, we've etched them. We've delivered what we've delivered here. We just haven't done a lot of the evaluation of the radial stiffness, and we're in the process of doing that. So watch this space, because we do think we have the nitinol, you know, stents very close to being developed. And the PhD student that I mentioned earlier, Sam Garrity, he's really working on now optimizing those designs and optimizing stents to minimize damage to arteries while still getting the vessel open as much as we need it to for coarctation.
Yeah, very interesting. And with that, how do the mechanical properties, how do they differ, I guess, to adults in terms of implanting stents? Does it have to be much lower radial force, or do you understand that yet?
Yeah, that's a good question, because actually the aorta itself is a much more elastic vessel than, say, for example, a more muscular coronary artery, where we see so many of the adult stents going in. And it's also much larger diameter. Even in a child, we're talking about vessels here of 10 to 15 millimeters, whereas, you know, coronaries are 3-4 millimeters. so quite different requirements. So other thing we are doing now is, given there's so little information on aortic coarcted tissue, is that we are taking neonates that have undergone surgery. So we've a new, one of our collaborations again with Professor Damien Kenny. We have an MD researcher, Niall Annan, who's just started working with us, and he's just to start in a week's time, but essentially he's from the clinical side, and he's going to take that tissue that comes from neonates when they're having the surgical reconstruction, and we're going to mechanically test it. And we're also going to look at kind of the composition of that tissue. How different is it to healthy aorta? Because there is almost nothing known about coarcted tissue. So while we're putting these stents in, it's often kind of looking at visually, do you get the expansion you need with a stent that wasn't designed for that purpose? So we need to know a lot more about the tissue we're putting these stents into. And that's one way to do it, is to get that material. Now, it is from neonates. It's not necessarily the ones that will have stenting. They typically have surgery. But again, if we can even just see compositional changes, can we see that, oh, maybe there's very low collagen, or maybe there's very high elastin, or there's, there's different changes. It would be great to see that, or even how variable it is in stiffness. Units are very similar, they're very different, and we think there's a lot of information to be gained from that.
And might finish on one last question. You showed 3D printed stents there, and you showed them deployed into physiological geometry. Have you actually looked at crimping down a stent, loading it and deploying it? And I guess how did it go?
We have only very preliminary data on that yet, because following the etching, we can kind of, it's springy, if you like. Do you know what I mean? You can compress it by hand and things like that, and you can compress in the radial compression to a certain extent, and it will recover a certain amount of deformation. But in terms of crimping it right down to go into a catheter, no, it would not have that level of recoverability at this point. And even the designs we have wouldn't be the most ideal for crimping. So we're working on better designs now. We wanted to start with something, as I said, that really conform to additive rules and then show flexibility in design. Now we need to go back and design with better, like, more of these flexible connectors and so on that enable it to be crimped down more easily. And we need to heat treat. So heat treatment does have to be done, to really achieve that kind of level of deformation crimping. And we are in the process of doing that at the moment.
Thank you very much. It's very exciting work, and I'd say everyone here would look forward to see how it progresses over the coming years.
Yeah, sure. Come back again for another biomedical division talk in a year's time. We see where we're at.
So with that, I'd like to thank Professor Tríona Lally for her talk and finish the session here. Thank you everyone for attending.